All right, so these T cells, everybody in agreement? Pretty cool. All right, very important. Now, how about those B cells? I don't like them as much as I do T cells.
You don't?
But now, hey, glad we got them, right? Okay, so B cells. Note the difference. Activated when the antigen fits the shape of its receptor. Note the difference? Did we have this with the T cell? No. So that is telling me those B cells are my humoral response. Because those B cells are going to be in the blood, okay, the plant, think about the blood, okay, and they're going to have a particular receptor on them, and if there's an antigen moving around, okay, and it fits into the receptor, that will activate that B cell. This is where I was like, wait, I couldn't really fully answer your question. I said maybe, but for the most part, okay, B cell activation requires that T helper cell.
Does that helper cell have anything to do with the shape of the B cell's receptor? So the B cell would just have a certain receptor and it would just wait and wait till that type of antigen came in?
Yep.
So we have a lot of B cells with a lot of different receptors?
Yep. If you've got a good immune system. Now, most of the time, activation requires the T cell. The T cell releases cytokines that can stimulate the B cell. That list that I just gave you for the cytokines, all right, that T cell, maybe it needs B cells to get activated, so it'll release the chemical that'll activate the B cell. Does that make sense? So, when the B cells get activated, the B cells, some of them might become a memory cell. What did we see with the T cells?
They all clone.
They always create the memory cell. Other B cells are going to differentiate into plasma cells, and those are going to produce and secrete really large globular proteins, and these are our antibodies or the immunoglobulins. The B cells are the humoral response. Blood is one of the humors of the body.
So B cells have the humoral immune response, but T cells have the
Ability to help activate them.
okay.
Now, the plasma cell, when the B cell differentiates into the plasma cell, they have now become basically this factory. So, antibodies, which can attach to an antigen, okay? Plasma cells secrete those. So this plasma cell can become a factory to make antibodies that we need to fight an antigen. Basically, this little itty bitty factory, and when we get infected, if this becomes activated, the number of antibodies that it can produce against an antigen is mind-boggling to fight that infection. Because we're now in the humors, the fluid of the body, the production that's made by these plasma cells, they're going to be found in the fluids of the body. And they will be specific. So whatever antigen needs to be engulfed, taken care of, gotten rid of, okay? The antibodies that will get produced are specific only for that antigen. The immunoglobulins get a little more interesting. I think in your book, let me see if I get into detail with those. Yeah, I've got a slide with the immunoglobulins. So we activate the B cell. B cell, some of them might become a memory cell. The other ones are going to become the plasma cells to produce the antibodies or the immunoglobulins. So, if we begin to see over here, they're starting out right here for number 1. I want to come over here, all right? That macrophage basically has encountered something, digested it, and now wants to display part of it on the surface. So it takes it, eats it up, and puts a little piece of it on its plasma membrane. Does everybody see that? Okay? Trying to see. Okay, so now, if we're having the macrophage display the little pieces of the pathogen, if we kind of look at what happens, that antigen, that little piece that gets displayed, remember how I said we would have the helper T cells? And what did we say was needed for the most part to activate the B cells? We need to have the T helper cell, okay? So that T cell can match that antigen. The T cell says, hey, hello. Now, do you remember that T cell, I said one of the things that it can produce are the cytokines, and there was a list of cytokines like the interferons and so forth. And then one of the type of cytokines were the interleukins. And the interleukins' job was to help help to activate other T cells, and then help the T cells to proliferate, increase in number, right? So my T helper cell, basically that T cell recognized the pathogen, it activated it, it needs to be a helper T cell, so that helper T cell can release cytokines, okay, which can go on to have different effects based on the list that we just discussed, okay? And it can go on to help generate more T cells. So the T cell, if we have that helper T cell become activated, so now if we have that helper T cell, we can get this cytotoxic T cell going. If we get the cytotoxic T cell going, we're going to eat stuff up. And in that case, for those T cells, we get the memories. Now, over here, if we created the cytokines, and those cytokines now activated the B cell. All right, look at this. I have a B cell, I have a B cell, and my B cells have on them receptor sites. Now, that T cell has activated this B cell. The B cell, that pathogen can now attach to these receptor sites. So it's picking up or fighting or getting to that pathogen. The B cell will begin to combine based on the receptor and the antigen. And we have now activated this B cell. Is everybody following me for the B cell so far? So we got presented, we activated the T cell. T cell can activate the B cell. Rebecca, you've got your eyes scrunched up.
Well, I'm waiting for you to finish because I'm wondering if when you're talking about the B cells, do they hold or secrete the antibody?
Once they begin to get activated, they become a factory for either an antibody or an immunoglobulin. So in this case, okay, if we look at that activated B cell, because the antigen can attach to the receptor site, that B cell now activated, it can become a memory cell, but then we're going to have to go on and it can become the plasma cell. Okay, so if we begin to activate that B cell, the B cell makes copies of itself, because if it makes copies of itself, it has the receptor, and if it has the receptor, it can pick up antigen, 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 pick up antigen. Is everybody following me? So, the B cells, they can become a memory cell. The plasma cells, they go on to either become the cells that release an antibody or an immunoglobulin. So each time we duplicate a B cell, so 2 becomes 4, 4 would become 8, 8 would become 16, all right? And each time, if it's this one, okay, they're showing that we're going to release antibodies, okay? Each time, it's going to be this antibody factory, and if it was immunoglobulins, it would be the immunoglobulin factory. So it's going to be producing the receptors. One of them that gets differentiated, they now become a memory cell.
I thought antibodies and immunoglobulins were the same thing. What's the difference between them?
Antibodies are going to be this particular shape, okay? So it's going to have the ability to connect to something that's only that shape. And this would be a response that's going to be needed for a short term. Now, types. Whoops. When we think about antibodies, they're going to float around in the blood, okay? Their job attached to them so we can destroy them. Immunoglobulins are a little more complicated, okay? Take a break, be back at 12:30.